Hi, I'm Alison Harrill. I'm currently a faculty member in the Regulatory Sciences program in the Department of Environmental and Occupational Health. I'd like to tell you a little bit today about my work, which really involves the interplay between genetics and drug and chemical exposure. And I hope that by the end of the talk that you'll have some sense of what I'm doing. I'm very interested in collaborations and in working with you, so please, if this interests you, take a look and reach out to me. So let me tell you a little bit about why I'm doing what I'm doing. The majority of my work to date has really focused on improving the science of pharmaceutical safety testing. Why do we want to do that? Well, major investments have gone into research and development in pharmaceutical development, but currently it's taking over $1 billion to get a new drug to market. And at the same time, we're really getting fewer and fewer drugs into the hands of the patients that need them. What we're finding is that as the industry has really improved upon the analysis of drug pharmacogenetics, really the science of drug safety is lagging behind. So currently, drug safety is becoming really the major bottleneck in drug development. And so what is really needed are new tools that can better predict safety risks to patients. Genetic variation can actually be a significant contributing factor to how patients respond to drugs, be it related to efficacy or adverse reactions. While some genetic factors are related to characteristics of drug metabolism, others are not, and they can be very difficult to predict. So one example of this is a drug called abacavir, which was developed by GSK to treat HIV infection. And essentially what happened is during the clinical trial phase of this development program for abacavir, they found that some people developed a very severe hypersensitivity reaction. Now, scientists at GSK actually found that these people who were susceptible to this hypersensitivity actually had a specific gene variant in a haplotype called HLA-B5701. And the association was so compelling that the FDA actually allowed this drug to go to market. However, it had to be coupled with a pharmacogenetic test for HLA-B5701. The problem is that in the clinic, there are a very small number of cases, and so oftentimes we don't have the statistical power to detect a genetic effect. And so what we need are model organisms that we can use to ethically test these drugs that we know to cause adverse drug reactions, where we can then come up with a pharmacogenetic basis. So one strategy to both improve hazard identification and identify the genetic basis is to use model organisms, such as the mouse. And what we can do is actually exploit the inherent genetic variation in the mice to enable these types of approaches. My team has demonstrated in a number of cases that using mouse populations can actually really fill this need. In one case, we evaluated an antibiotic that was under development by Pfizer until they found in clinical trials that this antibiotic was associated with elevations in liver biomarkers that could perhaps indicate a potential risk for liver damage. And so what we found when we used an approach that utilized 34 different inbred mouse strains is that there are some strains that were susceptible to the liver injury risk of this particular antibiotic. So what we found really is that had you used a mouse population approach, you could have detected the liver safety risk of this particular drug. Now, this type of approach is not limited to the liver. In fact, we conducted a study recently with a drug called DB289, which was under development to treat African sleeping sickness and had great efficacy for that particular indication. DB289 actually failed in an expanded phase 1 safety study where 6 healthy volunteers developed severe delayed renal insufficiency. So by using the same type of approach with 34 different inbred strains, as well as coupling this with a very sensitive biomarker of kidney injury called kidney injury molecule 1, we were able to detect the renal safety risk of DB289. So importantly, because the genetics of these models is very well characterized, we can also then study and identify the genetic basis of the safety risk. And so we call this type of approach translational pharmacogenetics. So the idea here is that we identify a safety risk in people, we then model that in mouse populations. We use the genetic characterization of the mice to figure out what genes or gene variants are actually conferring susceptibility. And then we validate that genetic risk marker in people. So one example of this is a study that we conducted where we looked at genetic susceptibility to acetaminophen-induced liver injury. So this study was actually done while I was a graduate student at the University of North Carolina at Chapel Hill, and was under the direction of Drs. Yvonne Rusin, David Thregill, and Paul Watkins. And so what we did were parallel studies, one in mice or in our mouse population, and one in the clinic where we looked at healthy volunteers. So what we found in the mice is that there were strains of mice that were resistant to the toxicity and strains of mice that were very susceptible. And in parallel in the clinic, what we found is when we gave these healthy volunteers the maximum therapeutic dose of acetaminophen, that there were some individuals who had elevations in liver biomarkers, which could suggest some hepatocellular injury, although it was very low level. What we then did is we mapped the toxicity phenotype to the mouse genome, and we identified that variation in a gene called CD44 was associated with increased risk of toxicity. Now this was in mice. What we then did is we took DNA from those study subjects enrolled in our clinical trial, and we validated that risk factor in people. So we found that variation in CD44 was associated with increases in liver biomarkers in the context of acetaminophen. Now, more recently, the Acute Liver Failure Study Group has looked at this same genetic risk factor in patients that have had severe liver injury due to acetaminophen exposure. And they found that this particular risk factor validated in people who had acetaminophen-induced liver injury or severe liver failure due to acetaminophen. With our older model using discrete classical inbred strains, we had a limited capacity to identify the genetic basis of injury or toxicity. And so we're now bridging to using newer models like the diversity outbred and the collaborative cross, because these are models that have been rationally designed to maximize the genetic variation available in the population and to randomize that genetic variation across the entirety of the mouse genome. These models were actually developed by the Complex Trait Consortium in collaboration with the Jackson Laboratory. We recently completed a study in the diversity outbred mice where we investigated liver injury due to epigallocatechin gallate, which is a component of green tea extract. Now, green tea extract has been associated with very severe liver injury in consumer populations taking this particular herbal supplement. So what we found in the diversity outbred, which is a very large population of mice that are genetically diverse, is that we can detect individuals who are completely resistant to the toxicity, as well as individuals who have very severe liver toxicity. So the difference here is not the dose that we gave, but actually the genetic background of the individual. We then collaborated with the Drug-Induced Liver Injury Network, who made available to us DNA samples from about 30 subjects who had experienced severe liver injury due to green tea extract. And we were able to show an association in the mice that we validated in the humans with two different genes that may confer an increased risk to toxicity due to green tea extract. In the next phase, we'll be continuing qualification of the diversity outbred population for identifying safety risks. And we're actually using a couple of different paradigms to do this. So in the first paradigm, we're looking at drugs that are on the marketplace that are associated with idiosyncratic or very rare liver toxicity. In the second set of studies, we're actually evaluating drugs that we've obtained from pharmaceutical partners. And these are drugs that actually failed during the clinical phase of testing due to increases in liver enzymes that are indicative of injury that were not predicted by the standard or classical safety paradigm. In addition, we're taking advantage of the unique genetic characteristics of the diversity outbred population to identify pharmacogenetic risk factors that might enable personalized prescribing of different medications. So by leveraging all of these data, we may be able to improve the science of not only hazard identification in the preclinical space, but also in personalized medicine. Taken together, this seemingly simple idea has the power to revolutionize the science of drug safety testing and to prevent patient harm by reducing the risk of adverse drug events.